There is something happening inside you right now. Not something dramatic, not something loud, something quiet, something patient, something that only begins when you stop trying. It started the moment you closed your eyes, the moment your breathing softened, the moment the world, with all its unfinished sentences and unanswered questions, loosened its grip on you. You may not feel it. Your jaw has relaxed. Your shoulders have lowered a fraction. Your hands feel heavier than they did an hour ago. Outside, the world is still awake somewhere. But inside you, a different kind of activity has begun. An ancient one. For most of human history, no one knew this was happening. Even neuroscientists, people who dedicate their lives to studying the brain, did not discover it until surprisingly recently. And when they did, many of them thought the data had to be wrong, because what they found sounded almost impossible. Your brain does not simply rest when you sleep. It transforms. It shifts into a state that is physically different from wakefulness. Spaces between your brain cells expand. A hidden network opens, and a clear fluid begins to move, slowly, rhythmically, through the delicate pathways of your mind. Washing, clearing, restoring. Right now, as you lie there, tiny rivers are flowing through the architecture of who you are. They are carrying away metabolic waste, clearing proteins that build up during the day, removing fragments of effort, stress, and thought. This process cannot happen properly while you are awake. In fact, your brain sacrifices this cleaning time every time you cut your sleep short. Tonight, we're going to move slowly through this hidden system. We're going to explore how scientists discovered it, why it only works in deep sleep, and what it might mean for memory, mood, even long-term brain health. And somewhere along the way, you may begin to feel something subtle but comforting. That sleep is not the absence of life. It is maintenance. It is protection. It is care. So let your body settle a little more. Let the outside world grow distant. And let's step quietly into the part of the night where your brain begins to clean itself. For most of human history, sleep was considered a kind of necessary emptiness. A pause. A gap in the story of a life where nothing of real consequence took place. Ancient philosophers puzzled over it with tremendous seriousness. Aristotle wrote at length about sleep, proposing that it occurred when vapors from digested food rose upward through the body, reached the heart, cooled and condensed there, and caused the senses to temporarily shut down. To him, sleep was a physiological consequence of eating, a kind of thermal regulation for the human machine. For centuries, this basic conception, that sleep was essentially the absence of wakefulness, a biological power-down mode, dominated the way people thought about it. Even in the modern era, well into the 20th century, sleep science was a relatively sparse and under-resourced field. The tools to study the sleeping brain were limited. You could not watch neurons fire in a sleeping animal in real time. You could not track the movement of specific molecules through living tissue while a subject was unconscious. You could record electrical activity from the scalp using electroencephalography, the EEG, and that alone had been illuminating enough to reveal that sleep was not uniform, that the sleeping brain produced recognizable patterns of electrical activity that changed across the night, that something structured and possibly meaningful was happening. But the precise mechanism, the why at a biological level, remained stubbornly elusive. The question that haunted sleep researchers for a long time was deceptively simple. Why do we need to sleep at all? Think for a moment about what sleep costs from an evolutionary standpoint. When you are asleep, you are unconscious. You are vulnerable in ways that you are not when you are awake. You cannot eat. You cannot respond quickly to a threat. You cannot care for your children, defend your resources, seek a mate, or perform any of the survival-critical behaviors that evolution has spent hundreds of millions of years optimizing. And yet, every single animal with a brain sleeps. Not most animals, all of them. Dolphins sleep with half their brain at a time, the other half staying awake to control breathing. Fruit flies sleep, and when you deprive them of sleep, they die more quickly. Migratory birds catch microsleep in flight. Even certain species of jellyfish, animals with distributed nervous systems more like nets than brains, exhibit rest states with the behavioral hallmarks of sleep. The universality is overwhelming. Whatever sleep is for, it is so fundamentally important that evolution has kept it in place across essentially every branch of the animal kingdom, even in the face of the enormous vulnerabilities it creates. That kind of universal conservation is a powerful biological signal. It says, this thing matters. This thing is not optional. This thing serves a purpose so basic that the cost of not having it exceeds the cost of unconsciousness. So what is the purpose? Several answers had been established before the breakthrough we will discuss tonight. Memory consolidation is real and well documented. During specific stages of sleep, the brain replays and strengthens the neural patterns that were formed during waking experience, solidifying the day's learning into more durable long-term storage. Synaptic homeostasis, the idea that sleep allows synaptic connections that have been strengthened during wakefulness to be systematically renormalized, preventing the nervous system from becoming saturated, is another serious hypothesis with growing support. Sleep regulates hormones. It supports immune function, processes emotional experience. It clears inflammatory markers from the blood. It restores depleted neurotransmitters. These are all real and meaningful functions. But in 2013, a neuroscientist named Ken Nedergaard at the University of Rochester published research that offered something that none of these other theories had provided: a specific, concrete, mechanistic explanation for why sleep itself, the actual unconscious state, not just the passage of time, was required for a critical biological function that the brain could not perform any other way. What Nedergaard and her colleagues found was that the sleeping brain physically opens itself up. The microscopic spaces between brain cells, spaces filled with a fluid called interstitial fluid, expand dramatically during sleep by as much as 60% compared to wakefulness. And into those expanded channels, a flood of cerebrospinal fluid rushes in, sweeping through brain tissue like a slow and purposeful tide, picking up molecular waste products that had accumulated during the waking day and carrying them out of the brain along well-defined drainage routes toward the body's lymphatic circulation. The brain was cleaning itself, not metaphorically, not poetically, literally, physically, chemically cleaning itself, rinsing out the metabolic debris of consciousness, flushing the byproducts of neural activity out of the tissue and into a disposal pathway that led ultimately to the liver, the kidneys, excretion. This was waste clearance on a brain-wide scale, and it happened during sleep because the physical changes necessary to enable it, the expansion of the extracellular space, the shift in fluid dynamics, could only occur when the brain's arousal systems were quiet. This was not a minor refinement of existing knowledge. This was a paradigm shift of the first order. It offered, for the first time, a mechanistically specific answer to one of biology's oldest questions. Why do we sleep? Among other things, because the brain must be cleaned, because the metabolic cost of consciousness is not only energetic but chemical, leaving behind molecular waste that must be systematically removed or it will accumulate to dangerous concentrations. Because the cleaning system that performs this function requires a brain that is asleep to operate at full capacity. And because the consequences of not cleaning the brain regularly enough are severe, real, and cumulative. The system responsible for all of this is called the glymphatic system. Its name was coined by Nedergaard and her colleague Jeffrey Iliff to honor two key features of its biology. It is managed by glial cells, specifically the astrocytes, the star-shaped supporting cells of the nervous system, and its function closely parallels the lymphatic system of the rest of the body. The glymphatic system was identified as a major, previously unrecognized pathway for brain-wide waste clearance, a system that had been operating every night in every sleeping mammal for the entirety of mammalian evolutionary history, undetected by science until the second decade of the 21st century. It is worth sitting with the strangeness of that for a moment. A system this significant, active every night in every sleeping human being on the planet, arguably one of the most important physiological processes in the human body, with direct relevance to some of the most devastating neurological diseases we know, went unrecognized until recently. Not because no one was curious, not because the technology was simply unavailable in any meaningful sense, but because no one was asking precisely the right question in precisely the right way, with precisely the right experimental tools, at precisely the right moment in the history of neuroscience. Science often works this way. The most profound discoveries are frequently not found by looking in new places, but by finally seeing clearly what was always there. The glymphatic system did not come into existence in 2012 when Nedergaard's team published their first paper on it. It has been running in your brain since the night you were born. What changed in 2012 was our ability to perceive it. There is also something quietly moving about the context of this discovery. Nedergaard has described how she came to the question of brain waste clearance through frustration. Frustration at the fact that despite years of productive research on astrocytes and their role in brain function, she could not find a direct, immediate clinical application for what she was learning. She kept asking herself, what practically does this knowledge do for patients? What mechanistically does the brain do with the waste that neurons generate? And it was that specific, patient, clinically motivated question that ultimately led to the discovery of a system whose clinical implications are enormous, potentially touching every condition from Alzheimer's disease to traumatic brain injury to stroke to normal aging. The most important scientific questions are often the most basic ones. And sometimes the most basic ones have been hiding in plain sight, active in the dark, waiting quietly for someone to look. Tonight we are looking. Let us follow the fluid into your sleeping brain and understand, in careful and gentle detail, what is happening there right now. To understand the glymphatic system, it helps first to appreciate the problem it solves. The human brain is, by a significant margin, the most metabolically active organ in the body. Though it represents only about 2% of total body weight in the average adult, it consumes roughly 20% of the body's total resting energy expenditure. This is a remarkable and disproportionate energy budget. The brain's metabolic rate is approximately 10 times higher than the average for the rest of the body's tissues. All of that energy consumption produces heat. It also produces waste. Neurons, in the constant and extraordinary activity of firing electrical signals, maintaining ion gradients across their membranes, synthesizing and releasing neurotransmitters, repairing their own structural components, and sustaining the molecular machinery of thought and feeling and memory, neurons generate a steady accumulation of metabolic byproducts. Some of these byproducts are small and diffuse easily. Others are large, complex proteins that do not clear themselves, that accumulate in the fluid surrounding the cells, and that, if allowed to remain at high concentrations, are toxic. The rest of the body handles metabolic waste using the lymphatic system, a vast network of vessels threaded throughout every organ and tissue, collecting excess interstitial fluid along with its dissolved waste products, filtering it through lymph nodes, and returning it to the bloodstream for processing. The lymphatic system is a kind of slow, pervasive drainage infrastructure that operates continuously in every part of the body, 24 hours a day, 7 days a week. But the brain was long thought to lack a lymphatic system. The blood-brain barrier, that highly selective membrane at the interface between the bloodstream and the brain's environment, strictly controls what enters and leaves the brain's tissue. And the standard teaching in neuroscience for most of the 20th century was that the brain had no conventional lymphatic drainage. The prevailing assumption was that metabolic waste simply diffused slowly through the interstitial fluid and was gradually absorbed into the cerebrospinal fluid that surrounds the brain. This assumption was, as it turned out, radically incomplete. Nedergaard's key insight came from a new way of looking at a structure that had been known for a long time: the perivascular space. Perivascular spaces are the narrow, fluid-filled gaps that exist between the outer wall of a blood vessel and the surrounding tissue. They had been noticed by anatomists in the 19th century and named for two of the researchers who described them. They are sometimes called Virchow-Robin spaces, but their function had never been fully characterized. They were generally considered to be simple anatomical spaces without a major physiological role. Nedergaard's team began injecting small fluorescent tracer molecules into the cerebrospinal fluid of living mice, and then using a technique called two-photon in vivo microscopy to watch what happened in real time. Two-photon microscopy is a form of optical imaging that uses infrared laser light to generate images of living tissue with extraordinary spatial resolution and minimal damage. It allows researchers to watch individual cells and molecular events inside a living, functioning organ, including the brains of mice that are awake or asleep. What they saw in those first experiments was that the fluorescent tracers they had injected into the CSF did not simply remain on the surface of the brain or diffuse uniformly through the tissue. They flowed, purposefully, rapidly, in a clearly organized pattern. They moved along the outside of the brain's arteries, following those vessels inward as they penetrated from the brain surface down into the deeper tissue. The CSF was entering the brain through a network of perivascular channels, traveling alongside the arteries like water flowing along the outside of a pipe. And when the researchers compared what happened in awake mice versus sleeping mice, the difference was immediate and dramatic. In awake animals, the fluorescent tracers barely moved into the brain at all. The perivascular channels appeared to be effectively closed, the fluid at the surface, the brain tissue a sealed interior. But almost immediately after the animals fell asleep, or after they were given anesthesia that simulated sleep-like brain states, the tracers flooded into the brain. The perivascular channels opened, the CSF poured inward along the arterial pathways, and the deep tissue of the brain was bathed in the incoming fluid. The follow-up question was obvious. Where did the fluid go after it entered the brain? Tracing it further, the researchers found that the incoming CSF mixed with the interstitial fluid of the brain parenchyma. The complex, molecule-rich fluid that fills the microscopic spaces between individual brain cells. In that mixing, the CSF took up dissolved substances from the interstitial fluid, including metabolic waste products. And then the now mixed fluid was directed toward the paravenous spaces, the channels surrounding the brain's veins, and from there it drained outward. The venous paravascular spaces acted as drainage channels, carrying the mixed fluid out of the brain's interior toward the meninges and eventually toward lymphatic vessels that exit the skull. A complete circuit, a hydraulic system, inward along arteries through the tissue, outward along veins. Clean fluid in, waste-laden fluid out. This system was named the glymphatic system, from glial, for the astrocytes that form the channel walls, and lymphatic, for its functional resemblance to the peripheral lymphatic system. The naming was precise and meaningful. It acknowledged both the cellular machinery that enables the system and the systemic function it serves. The brain, it turned out, did have a lymphatic system, just not one that anyone had recognized before. The key cellular player, the reason the GL prefix was chosen, is the astrocyte. Astrocytes are one of the most abundant cell types in the brain, roughly matching neurons in number, and they are increasingly understood to be active participants in brain physiology rather than passive scaffolding. They are shaped like stars. The name comes from the Greek astron, for star, with long, branching processes extending in many directions. Some of those processes wrap around neurons and synapses, where they play roles in regulating neurotransmitter levels and supporting synaptic function. Other processes reach toward blood vessels, ending in flattened, paddle-like structures called end feet that fit snugly against the outer walls of the capillaries and arteries. The end feet of astrocytes are densely populated with a specific type of water channel protein called aquaporin 4, often abbreviated AQP4. Aquaporins are among the most elegant molecular machines in biology, tiny protein channels, just a few nanometers wide, that span the cell membrane and allow water molecules to pass through in single file at remarkable speed. Without aquaporins, water can still cross cell membranes, but slowly through diffusion. With aquaporins, water moves rapidly, specifically, and in a controlled, directional manner. Aquaporin 4 is the most prevalent water channel in the brain, and its distribution is not random. It is concentrated specifically in the astrocyte end feet, the surfaces that face the paravascular spaces. This orientation is crucial. It positions the water channels precisely at the interface between the paravascular space and the brain's interior, allowing fluid to move rapidly from the CSF flowing in the periarterial channels into the interstitial space of the brain tissue. In mice that have been genetically engineered to lack the AQP4 gene, glymphatic clearance of large proteins is reduced by approximately 70% compared to normal animals. The aquaporin 4 channels are not a minor modifier of glymphatic flow. They are essential to its operation. The discovery and characterization of the glymphatic system, and particularly the demonstration that it clears amyloid beta, one of the key proteins implicated in Alzheimer's disease, at a rate far higher than any previously known mechanism, was recognized as one of the top scientific breakthroughs of 2013 by Science magazine. In 2024, Dr. Nedergaard was awarded the Nakasone Award by the International Human Frontier Science Program Organization for her groundbreaking discovery and exploration of the glymphatic system. And in the same year, for the first time, researchers at Oregon Health and Science University directly visualized the glymphatic pathway in living human patients undergoing brain surgery, watching cerebrospinal fluid flow through perivascular channels in human brain tissue, confirming in people what had been established in rodents. The brain cleans itself in sleep. This is no longer a hypothesis. It is an observed, measured, visualized fact. Now let us trace the path of the river in greater detail, because understanding the mechanics of how the fluid moves is the first step to appreciating the profound engineering of what happens inside you every night. Close your eyes, if you have not already, and imagine something like an underground spring. Somewhere beneath a quiet hillside, water filters through layers of rock and soil. It has no turbines, no pumps, no machinery to drive it forward. It moves because of pressure and gravity and the shape of the channels nature has carved for it. It is patient, persistent, thorough. Your brain has something very like this. The story of the glymphatic river begins in the deepest interior of your brain, in a series of interconnected, fluid-filled chambers called ventricles. There are 4 ventricles in the human brain. 2 large ones in the cerebral hemispheres called the lateral ventricles, and 2 smaller ones deeper in the brain structure. These ventricles are lined with specialized cells, and nestled within them are intricate, richly vascularized structures called the choroid plexus. The choroid plexus is the spring. It is where cerebrospinal fluid originates. The choroid plexus consists of specialized epithelial cells, cells that line the interface between the blood supply and the brain environment, that actively filter blood plasma. They take up water, ions, glucose, and certain proteins from the bloodstream and combine them in carefully controlled proportions to produce a clear, almost protein-free fluid, the cerebrospinal fluid. Your choroid plexus produces approximately 500 milliliters of CSF every single day, about the volume of a standard water bottle. And since the total volume of CSF in the brain and spinal cord at any given moment is only about 150 milliliters, the entire fluid volume turns over completely about 3 to 4 times every 24 hours. This is a system of continuous production, continuous flow, and continuous replacement. Not a static pool, but a living current. From the ventricles, the freshly produced CSF flows outward through a series of narrow connections between the chambers, through an opening called the foramen of Magendie and 2 openings called the foramina of Luschka, and emerges into the subarachnoid space. The subarachnoid space is the fluid-filled gap between 2 of the membranes that enclose the brain, the outer arachnoid membrane and the inner pia mater, which clings directly to the surface of the brain tissue. In this subarachnoid space, the CSF forms a protective cushioning layer around the entire brain and spinal cord. It is the fluid you see on brain MRI images as the bright white space surrounding the brain, a hydraulic shock absorber that protects the most sensitive tissue in the body. But the CSF in the subarachnoid space does more than cushion. As Nedergaard's research revealed, it flows. It enters the brain. It dives inward along channels that run alongside the arteries, the blood vessels that carry oxygenated blood from the surface down into the brain's interior. These are the periarterial spaces, sometimes called the Virchow-Robin spaces, and they are the entry point of the glymphatic river into brain tissue. The movement of CSF into the brain along the periarterial channels is driven by several forces working together. There is the pressure gradient generated by the continuous production of CSF at the choroid plexus, the spring pressure that pushes the fluid outward and inward. There is the pulsatile expansion and contraction of the arteries with each heartbeat, which transmits a pressure wave outward into the perivascular space and nudges the fluid forward. And, as groundbreaking research in 2025 revealed, there is the rhythmic, slow constriction and dilation of the arteries driven by oscillations in a neurochemical signal during deep sleep, a mechanism we will explore in detail shortly. The CSF moves along the periarterial channels, driven by these forces, flowing deeper into the brain as the arteries penetrate toward its center. At the level of the smallest vessels, the capillaries that reach every corner of the brain parenchyma, the perivascular space is narrow. Here, the astrocyte end feet and their dense coating of aquaporin-4 water channels form a thin boundary layer between the perivascular channel and the interstitial space of the brain tissue. Driven by the pressure differential and the osmotic dynamics at the AQP4 channels, the CSF passes through this boundary water molecules threading through the aquaporin channels into the extracellular space of the brain itself. The interstitial space is the microscopic geography between individual brain cells, the interconnected labyrinth of narrow channels, filled with a carefully balanced fluid that bathes every neuron and astrocyte and oligodendrocyte and microglial cell in the brain. It is not empty. It contains a complex mixture of ions, signaling molecules, growth factors, metabolic byproducts, and the byproducts of neural activity, including the proteins that are the targets of the glymphatic clearance system. When the incoming CSF mixes with the interstitial fluid, a process of exchange takes place. The metabolic waste products dissolved in the interstitial fluid are taken up by the incoming fluid, carried along with the flow as the CSF mixes and moves through the extracellular space. The flow is directional, from the periarterial side toward the perivenous side, sweeping through the tissue in a slow, steady current driven by the overall pressure gradient of the system. When the waste-laden fluid reaches the venous side of the brain's vascular tree, the veins and venules that carry deoxygenated blood back toward the heart, it enters the perivenous spaces. These are the drainage channels of the glymphatic system, the riverbed through which the cleaned-out interstitial fluid exits the brain's interior. From the perivenous spaces, the fluid moves outward to the meninges, and from there it takes several routes out of the skull. Some of it follows the sheaths of the cranial nerves, thin, fluid-filled channels that surround the nerves as they exit the brain. The olfactory nerves, which pass through the cribriform plate at the base of the skull into the nasal cavity, are one of the primary exit routes. From the nasal submucosa, the fluid enters the lymphatic vessels of the neck and eventually the general circulation, where the waste proteins it carries are delivered to the lymph nodes and ultimately to the liver and kidneys for processing and elimination. Other fluid reaches the meningeal lymphatic vessels, a network of lymphatic channels discovered in 2015 running along the dura mater, the outermost membrane of the brain. These meningeal lymphatics drain toward the deep cervical lymph nodes, providing another pathway for protein clearance from the brain. The entire circuit, from the choroid plexus to the subarachnoid space to the periarterial channels to the interstitial space to the perivenous channels to the meningeal lymphatics to the cervical lymph nodes, is a complete, purposeful hydraulic loop. A brain-wide waste management system of extraordinary sophistication. What makes it particularly beautiful from a biological engineering standpoint is that it uses the brain's existing vascular architecture as its scaffolding. The same blood vessels that bring oxygen and glucose to neurons also create, along their outer surfaces, the channels through which CSF flows. The glymphatic system is not a separate structure that happens to exist in the brain. It is built into the brain's basic tissue organization, woven around the vasculature, enabled by the astrocytes that already surround every blood vessel, using a simple but elegant trick, the aquaporin for water channels, to move fluid efficiently through a tissue that would otherwise resist fluid flow. Evolution, working over hundreds of millions of years, found a solution to the problem of brain waste clearance that required no new organs, no new vessels, no separate infrastructure. It used what was already there. It simply added water channels at precisely the right location, oriented in precisely the right direction, and created a pressure system that, when the conditions of sleep allowed it, would drive a river of cleaning fluid through the most sensitive tissue in the body. You are carrying this river with you right now. It runs inside you every night. And right now, if you have settled deeply enough into sleep, it is running. Sleep is not a single state. This is one of the first and most fundamental things that sleep science established, beginning with the pioneering EEG recordings of the mid-20th century. What we call sleep is actually a structured sequence of distinct states, each with its own characteristic pattern of brain activity, its own hormonal environment, its own specific biological functions. Understanding the different stages of sleep is essential to understanding the glymphatic system, because not all stages of sleep are equally effective at driving brain cleaning. One stage, in particular, is far more important than all the others. When you first lie down in the dark and close your eyes, your brain transitions through the earliest stages of sleep. Your alpha waves, the oscillations associated with relaxed, wakeful rest, begin to slow and diminish. You enter stage 1, the lightest sleep, a brief, easily disrupted state in which you are technically asleep, but from which the smallest sound or movement can easily wake you. You might experience a sudden jerk, the hypnic jerk as researchers call it, as your body crosses the threshold from wakefulness. Your muscles twitch. Your eyes roll slowly behind their lids. From there, if undisturbed, you drift into stage 2. Here the brain begins to produce characteristic bursts of synchronized electrical activity called sleep spindles, rapid oscillations of 11 to 16 cycles per second that last for about half a second to 2 seconds each, appearing as distinctive bursts on an EEG recording. These sleep spindles appear to play a meaningful role in protecting sleep. They suppress the brain's responses to incoming sensory signals, making it less likely that environmental noise will wake you. They are also associated with memory consolidation, with the offline processing of newly acquired information. Sleep spindles are one of those features of the sleeping brain that seem almost too elegant to be accidental. They appear at exactly the right moment in the sleep cycle, serve multiple purposes simultaneously, and are so precisely timed and structured that they read under the EEG almost like a musical signature, the brain playing a specific recognizable theme. But the stage that matters most for the glymphatic system comes next. Stage 3, deep slow-wave sleep, also called N3. This is the deepest stage of non-REM sleep, and it is characterized by the large, slow electrical waves called delta waves, oscillations of 0.5 to 4 cycles per second, with amplitudes much larger than the faster rhythms of lighter sleep. If you imagine the EEG of a sleeping brain as a kind of musical score, stage 3 is the long, slow, sustained notes of a cello rolling through the brain in measured waves, while the lighter stages are more like the faster, more intricate melodies of the same composition. During this stage of sleep, something remarkable and specific happens to the physical structure of your brain. The microscopic architecture of the brain's extracellular space changes. The story of that change begins with norepinephrine, a neurochemical that you probably know as a close cousin of adrenaline, involved in alertness, attention, and the body's arousal response. During wakefulness, a small but powerful cluster of neurons in the brainstem called the locus coeruleus produces norepinephrine at a steady level and releases it throughout the brain. This tonic release of norepinephrine is a signal of waking awareness. It contributes to the vigilance and attentiveness of the conscious state. And it also, through mechanisms involving the astrocytes and their regulation of ion transport, keeps the brain's interstitial space in a compressed state. The cells slightly swollen relative to their sleep state size, the extracellular channels narrow. When you fall asleep, and particularly as you descend into stage 3 deep sleep, the locus coeruleus quiets. Norepinephrine levels in the brain drop significantly. And as they drop, something opens. The astrocytes, no longer receiving the norepinephrine signal that kept them slightly inflated, allow water to redistribute across their membranes. The ions that regulate cell volume shift, and the cells of the brain subtly shrink, not dramatically, not visibly, but enough to significantly widen the extracellular channels between them. The interstitial space expands. In the landmark 2013 study by Nedergaard's team, this expansion was measured directly in living mice using molecular diffusion measurements, and the finding was striking. During sleep, the interstitial space was approximately 60% larger than during wakefulness. 60%. Not a marginal difference. A transformation. The narrow alleys between the cells of your sleeping brain become significantly wider channels, and the resistance to fluid flow through those channels drops accordingly. The glymphatic system, which could only force a trickle through the compressed tissue of the waking brain, now has room to run. The river can flow. But the story of how the river is actually driven through the tissue became even more fascinating in 2025, when Natalie Haugland, Maiken Nedergaard, and their colleagues published a study in the journal Cell that represented, by many accounts, the most detailed characterization of the glymphatic pump mechanism to date. The researchers used an array of sophisticated technologies, fiber photometry, engineered genetically encoded sensors for norepinephrine, electrophysiology, CSF tracer imaging, and optogenetics to study the sleeping mouse brain with unprecedented precision. What they found was that the locus coeruleus, during NREM sleep, does not simply go silent. It oscillates. The norepinephrine it releases during deep sleep fluctuates in slow, rhythmic waves, at a frequency of approximately once every 50 seconds, an infraslow oscillation far below the frequencies of brain activity during wakefulness. These norepinephrine waves, rising and falling with gentle regularity, drive a corresponding rhythm in the brain's blood vessels. Norepinephrine is a vasoconstrictor. It causes blood vessel walls to contract. When the norepinephrine signal rises, the arteries of the brain tighten slightly. When it falls, they relax and dilate. The result is a slow, rhythmic constriction and dilation of the cerebral arteries, occurring at the same frequency as the norepinephrine oscillations, roughly once per minute. This slow arterial rhythm is called vasomotion. Vasomotion, the researchers found, acts as a peristaltic pump for the glymphatic system. Think of how a peristaltic pump works. It squeezes a flexible tube in a rhythmic wave, and the sequential compression drives fluid through the tube. When a brain artery constricts, its outer wall presses slightly outward into the periarterial space, the channel where CSF flows alongside the vessel, creating a gentle pressure that drives CSF forward along the channel, deeper into the brain. When the artery dilates, the channel widens again, drawing more CSF in behind it. The slow, rhythmic constriction and dilation of the arteries, driven by those infraslow norepinephrine oscillations, drives the cerebrospinal fluid through the glymphatic channels in a directed, purposeful flow. This is the pump. This is the mechanism that drives the cleaning river through the sleeping brain. Not the heartbeat, though cardiac pulsation contributes some pressure. Not mere osmotic gradients, though those matter too. But the slow rhythmic vasomotion, driven by the oscillating norepinephrine from the locus coeruleus, acting as a peristaltic pump on the periarterial channels of the glymphatic system. The researchers put it beautifully. It is like turning on the dishwasher before you go to bed and waking up with a clean brain. The 2025 study also made a finding with significant practical implications. The team examined the effects of a common sleep medication, zolpidem, sold under brand names including Ambien, on glymphatic clearance in mice. They found that zolpidem suppressed the infraslow norepinephrine oscillations that drive vasomotion, and that this suppression reduced glymphatic clearance by roughly 30% compared to natural, undrugged sleep. The animals technically fell asleep. By most measures, they appeared to be sleeping. But the slow-wave architecture of their sleep was subtly altered. The oscillating norepinephrine was disrupted, and the pump ran at reduced capacity. This finding does not mean that sleep medications are broadly harmful, or that anyone who takes a sleep aid is damaging their brain. The relationship between specific medications, specific sleep architectures, and long-term brain health is complex and depends enormously on individual circumstances. But it underscores something that sleep researchers have been saying for a long time. Not all sleep is equivalent. The quality of sleep, specifically the depth of it, the completeness of the slow-wave architecture, the presence and regularity of those deep N3 oscillations, determines how effectively the glymphatic system operates. You cannot substitute shallow sleep for deep sleep and expect the same cleaning result. The river needs the pump. The pump needs the waves. The waves need the locus coeruleus to oscillate in its slow, infraslow rhythm. And that rhythm needs deep sleep, which means that everything you do to support deep, restorative sleep is also, specifically, something you are doing for your glymphatic system. The consistent sleep schedule that reinforces your circadian rhythm, making it easier to achieve the deep sleep stages. The cool, dark bedroom that signals to your brain that night has arrived. The evening routine that allows your arousal systems to settle before you lie down. The absence of screens and bright light in the hour before sleep, preventing the artificial blue wavelength stimulation that delays melatonin release. These are not vague lifestyle recommendations. They are specific interventions in support of a specific biological mechanism, with a specific and important biological outcome. Right now, if you have settled deeply enough, the oscillations are running. The norepinephrine is rising and falling in its slow, 50-second rhythm. Your arteries are pulsing gently in their clean, regular vasomotion. And the cerebrospinal fluid is moving. Now that we understand the mechanism, how the river is built, and how it is driven, let us consider what it carries. Let us look at the specific molecular contents of the brain's interstitial fluid, and why the nightly clearing of those contents matters so deeply for long-term brain health. The brain produces a continuous stream of metabolic waste products as a natural, inevitable consequence of being active. The vast majority of these are small, quickly diffusing molecules that pose no particular problem: carbon dioxide, lactic acid, small ions, ordinary byproducts of cellular energy metabolism. These clear easily through multiple mechanisms without requiring any dramatic intervention from the glymphatic system. But among the larger, more troubling molecules that accumulate in the interstitial fluid, two proteins have attracted the most intense scientific attention. You have almost certainly heard of them because they are the proteins most directly associated with Alzheimer's disease. They are amyloid beta and tau, and understanding what they are and why the brain needs to clear them regularly is essential to understanding why the glymphatic system matters. Amyloid beta is a small peptide, a short chain of amino acids, that is produced by neurons as a normal byproduct of their activity. It is cleaved from a larger protein called the amyloid precursor protein, which is embedded in neuronal membranes. This cleavage happens continuously in healthy brains. Amyloid beta is generated at a steady rate by neurons throughout the day and night. In a healthy, young brain with functioning glymphatic clearance, the rate of production is balanced by the rate of clearance, and the concentration of amyloid beta in the interstitial fluid remains relatively low and stable. But amyloid beta has a troublesome tendency. When it accumulates, when the balance between production and clearance tips even slightly in favor of accumulation, it tends to aggregate. Individual amyloid beta molecules stick to each other, forming small clusters called oligomers, which can then grow into longer structures called fibrils, which can then deposit as the amyloid plaques that are the most visible pathological hallmark of Alzheimer's disease. These plaques form preferentially in the spaces between neurons, where they disrupt synaptic function and trigger inflammatory cascades that damage the surrounding tissue. Research has established that more than half of the amyloid beta that leaves the brain under normal conditions exits via the glymphatic system. It is swept up by the flowing CSF and carried out along the paravenous drainage routes, ultimately reaching the cervical lymph nodes and the bloodstream, where it is processed by the liver. When the glymphatic system works efficiently, amyloid beta is cleared before it can accumulate to the concentrations where it begins to aggregate. When glymphatic function is impaired, the balance tips and accumulation begins. The clinical evidence for this relationship in humans is compelling. In 2018, researchers at the National Institutes of Health used positron emission tomography brain imaging to measure amyloid beta burden in the brains of 20 healthy adult volunteers before and after a night of sleep. The finding was striking and alarming in equal measure. Just one night of total sleep deprivation, a single night, produced a measurable increase in amyloid beta burden in the right hippocampus and right thalamus. Both of these regions are areas of established relevance in Alzheimer's disease. The increases were visible in the PET images as elevated radiotracer signal. One night without sleep, a measurable molecular change in the brain. This is not a catastrophic accumulation that dooms anyone to disease. The brain has enormous resilience, and a single disrupted night is followed by clearance that largely restores the balance. But the rapidity and sensitivity of the response tells us something important. The system is finely balanced, and sleep is a critical variable in maintaining that balance on a daily basis. Tau is the second protein of major concern. Tau normally plays an important structural role inside neurons, It associates with the microtubules, the internal skeletal structures of the neuron, helping to stabilize them and allowing the efficient transport of molecular cargo along the length of the cell. In a healthy neuron, tau is regulated, organized, and functional. But under conditions of stress, including the stress of excessive accumulation due to inadequate clearance, tau can become abnormally modified, particularly through a process called phosphorylation. Abnormally phosphorylated tau detaches from microtubules, aggregates with other tau molecules, and eventually forms the neurofibrillary tangles that are the second major pathological signature of Alzheimer's disease. The tangles form inside neurons rather than outside them, making them in some ways even more directly damaging to neural function than amyloid plaques. And their distribution in the brain follows a characteristic pattern, beginning in specific regions and spreading over years, that correlates with the progression of cognitive symptoms in Alzheimer's disease. A landmark 2019 study published in Science by Jera Holth and colleagues at Washington University in St. Louis demonstrated that tau, like amyloid beta, is regulated by the sleep-wake cycle. In mice, measurements of tau in the brain's interstitial fluid showed that levels were approximately twice as high during wakefulness as during sleep. The implication was clear. Sleep was promoting the clearance of tau from the interstitial space, and wakefulness was allowing it to accumulate. When the same research team looked at cerebrospinal fluid samples from human volunteers who had undergone one night of total sleep deprivation, they found something even more sobering. Tau levels in the CSF increased by approximately 50%, a larger relative increase than had been seen for amyloid beta. One night of lost sleep in young, healthy adults produced a 50% increase in tau protein in the brain's fluid environment. The picture that emerges from this body of research is consistent and serious. The glymphatic system clears amyloid beta and tau on a nightly basis. Sleep is required for optimal glymphatic function. Sleep deprivation acutely impairs that clearance. And the proteins that accumulate when clearance is impaired are the same proteins that define the pathology of the most common neurodegenerative disease of aging. This does not mean that Alzheimer's disease is simply a disease of too little sleep. It is not that simple. Alzheimer's pathology is multifactorial, with genetic susceptibilities, vascular contributions, metabolic factors, and environmental variables all playing meaningful roles. And the relationship between sleep and Alzheimer's is bidirectional. The disease itself disrupts sleep architecture, creating a vicious cycle in which pathological accumulation impairs sleep, and impaired sleep further promotes pathological accumulation. But the sleep glymphatic protein accumulation axis is a real and meaningful part of the story, and one that is rapidly becoming a target of active therapeutic investigation. Beyond amyloid beta and tau, the glymphatic system clears other metabolically important molecules from the brain's interstitial fluid. Alpha-synuclein, the protein that aggregates in Parkinson's disease, is also present in the interstitial fluid and is cleared via glymphatic routes. Various inflammatory mediators, small metabolites of energy metabolism. The precise inventory of everything the glymphatic system clears is still being compiled, but the general principle holds across all of them. The interstitial fluid of an active brain accumulates the molecular residue of consciousness, and the sleeping brain's glymphatic system is the mechanism by which that residue is regularly removed. There is something almost poignant about this. The idea that every thought you have, every sensation you feel, Every decision you make leaves behind a microscopic molecular signature in the fluid between your neurons. Consciousness is expensive. It leaves a trail. And sleep is the nightly accounting that settles the bill, erases the trail, and prepares the molecular stage for another day of experience. The river carries away the residue of who you were today, and what flows back in is the clear fluid of tomorrow. There is something worth mentioning as a quiet aside before we continue. Something that may make you think just briefly about how you are lying right now. Not to create anxiety, and not because changing your position is urgent or required, but because this small piece of science is genuinely interesting and, for some of you, may feel like a gentle confirmation of something your body already knew. In 2015, a research team at Stony Brook University in New York published a study in the Journal of Neuroscience that examined whether body position during sleep affects the efficiency of glymphatic clearance. The researchers were led by Hidek Lee and Helene Benvenisti, with collaboration from Maiken Nedergaard's group at the University of Rochester. Using dynamic contrast-enhanced MRI, a technique that tracks the movement of a contrast agent injected into the CSF, the team was able to visualize and quantify glymphatic transport in the brains of anesthetized rodents in 3 different positions: lying on their backs, lying on their fronts, and lying on their sides. The results were consistent and striking. Glymphatic transport was most efficient in the lateral position, on the side. When the animals lay on their sides, the MRI images showed greater penetration of the CSF tracer into the brain tissue and more efficient clearance of amyloid beta compared to both the supine and prone positions. The prone position, lying face down, was actually the least efficient for glymphatic clearance, with tracer molecules showing more retention in the tissue and slower overall flow. Why would position matter? The researchers proposed several possible mechanisms. Gravity obviously affects fluid distribution in the body and in the brain. The direction of the gravitational vector relative to the anatomy of the perivascular spaces would influence the ease of fluid flow in different directions. The geometry of the skull, the arrangement of the major venous drainage structures, and the relationship between the brain and the surrounding meningeal layers all differ depending on body position, and any of these could influence the pressure dynamics of the glymphatic system. The precise mechanistic details of why lateral sleeping appears superior for glymphatic clearance are not yet fully worked out. This is an area of active investigation. Dr. Nedergaard made a comment about this finding that is worth pausing on. She noted that lateral sleeping is already the most common sleeping position in humans and in most other mammals, including animals in the wild. In evolutionary terms, sleeping positions are not random, and the fact that the most widespread natural sleeping posture in mammals also appears to be the most efficient for brain waste clearance is at minimum a provocative coincidence, and quite possibly a genuine evolutionary adaptation. Bodies that slept on their sides may have had better glymphatic clearance, accumulated less amyloid beta over a lifetime, maintained better cognitive function into old age, and, in terms of evolutionary fitness, had better outcomes as a result. We carry, in our preferred sleeping positions, possibly millions of years of evolutionary optimization. The body already knows, at some level, what the science is only now beginning to articulate. One thing worth noting: this research was conducted in rodents, and direct confirmation in humans awaits further investigation. The anatomy of human sleep and the specific details of how body position affects CSF dynamics in the human brain may not translate exactly from rodent studies. The researchers themselves were careful to note this limitation. What seems reasonable to conclude is that sleep position is a real variable in glymphatic efficiency, that it matters, but that the specific quantitative benefits in humans remain to be precisely characterized. There is also no clear scientific consensus on whether the left side or the right side is preferable. Various anatomical arguments have been made for one or the other: the arrangement of the major veins, the position of the inferior vena cava, the asymmetry of certain lymphatic drainage structures, but the neuroscience literature does not currently support a strong preference for either specific lateral position in terms of glymphatic function specifically. Both sides appear to be superior to the back and front positions for the purposes of the glymphatic system. If you happen to be lying on your side right now, there may be a small, quiet comfort in knowing that your body found this position naturally, that what feels comfortable is, at least potentially, also what supports the work happening inside your brain. Your body and evolutionary wisdom may be quietly cooperating. And if you are not on your side, if you are on your back or propped with pillows, or in some arrangement that simply feels right for your body tonight, please hold that without any anxiety. The most important variable in glymphatic function is not position. It is the depth and quality of sleep itself. A night of deep, restorative sleep in any comfortable position is vastly more beneficial than a night of restless, shallow sleep in the theoretically optimal lateral position. Your comfort and your ability to relax fully into sleep matters most. Position is a fine adjustment. Depth is the essential thing. Let your body settle into whatever position feels right. Let the tension in your muscles soften a little more. And let us continue. Understanding the costs of sleep deprivation is not about creating worry. You are already here, already resting, already doing exactly what your brain needs you to do. But understanding the costs illuminates the value. Knowing what the absence of something produces helps us appreciate, with more than intellectual acknowledgement, what its presence provides. And the costs of inadequate sleep, seen through the lens of the glymphatic system and the broader neuroscience of sleep, are worth knowing about clearly, calmly, and without exaggeration. When sleep is disrupted, shortened, or chronically insufficient, the glymphatic river slows. The 60% expansion of the interstitial space that deep sleep enables does not occur to its full extent. The infraslow norepinephrine oscillations that drive vasomotion and pump CSF through the periarterial channels are disrupted or absent. The aquaporin 4 channels in the astrocyte end feet, which depend on proper sleep architecture to maintain their functional polarization at the perivascular surfaces, become less efficiently organized. The net result is reduced glymphatic clearance. Less fluid moving through the brain, less waste being removed. The immediate molecular consequences are measurable. As the studies we discussed earlier demonstrated, even a single night of sleep deprivation produces detectable increases in amyloid beta and tau protein levels in the brain's fluid environment. These are not catastrophic. The brain has significant resilience, and one bad night followed by good sleep allows considerable recovery. But repeated nights of insufficient sleep create a cumulative deficit. The amyloid beta that accumulates when clearance is impaired does not simply wait patiently until better sleep arrives. It begins to aggregate, slowly over time at concentrations that may not yet cause any obvious symptoms. The balance tilts. The functional consequences of impaired glymphatic clearance and of sleep deprivation more generally are felt in daily life in ways that most of us recognize well, even without knowing their biological basis. Consider attention. The brain's capacity to maintain sustained, directed attention is one of the most sleep-sensitive cognitive functions. After a poor night's sleep, you know the experience. The difficulty holding a thread of thought, the tendency for the mind to drift, the increased effort required to do things that normally feel automatic. Research using sensitive cognitive testing has shown that even modest sleep restriction, reducing sleep from 8 hours to 6 hours per night for 2 weeks, produces cumulative cognitive deficits equivalent to 2 full nights of total sleep deprivation, and that subjects often underestimate how impaired they are. The subjective sense of how sleepy you feel stops matching the objective degree of cognitive impairment after about 10 days. Part of this impairment reflects the disruption of the molecular environment within which neurons operate. When the interstitial fluid carries higher concentrations of metabolic waste, when amyloid beta and inflammatory molecules are not efficiently cleared, the chemical conditions at synapses are altered, subtly but measurably. Synaptic function is exquisitely sensitive to its molecular environment, and even small deviations from the precisely balanced conditions of a well-rested brain may affect the speed and efficiency of neural processing. The emotional consequences of poor sleep are, for many people, even more immediately disruptive than the cognitive ones. Sleep deprivation dramatically increases emotional reactivity. The amygdala, the brain's threat detection and emotional processing center, shows markedly elevated responses to emotionally negative stimuli in sleep-deprived individuals. At the same time, the functional connectivity between the amygdala and the prefrontal cortex, the region associated with rational control, perspective-taking, and the regulation of emotional responses, is significantly reduced. In concrete terms, when you are sleep-deprived, your emotional reactions fire more intensely, and your capacity to modulate, contextualize, and rationally respond to those reactions is diminished. Small frustrations feel larger. Provocations that a well-rested version of you would handle with equanimity instead feel overwhelming. The emotional equilibrium that feels so natural when you are rested, the ability to feel something strongly and then return to center, to choose a thoughtful response over an impulsive one, becomes much more difficult to maintain. This is not a character weakness. It is a physiological state. It is a brain operating in a chemically suboptimal environment, with its regulatory circuits running at reduced capacity, in a context where the molecular clarity that sleep normally provides has been compromised. There is also a longer arc to consider, one that reaches across years and decades rather than days. Research has consistently found associations between chronic sleep disruption and accelerated cognitive decline. Longitudinal studies have shown that people who habitually sleep less than 6 hours per night have elevated risks of cognitive impairment in later life compared to those who sleep 7 to 8 hours. Brain imaging studies have found that poor sleep is associated with higher amyloid burden in the brain, a relationship that holds even in cognitively healthy middle-aged adults, decades before any obvious symptoms might appear. The relationship between sleep and Alzheimer's disease in particular is one of the most active research areas in neuroscience today. As we discussed, it is bidirectional and complex. Alzheimer's pathology itself disrupts sleep, creating a cycle that is difficult to untangle. But the glymphatic hypothesis offers a plausible mechanistic basis for at least part of the association. Decades of chronic sleep impairment, each night shortchanging the brain's cleaning cycle, could contribute to a gradual accumulation of the very proteins whose clearance sleep is supposed to ensure. None of this is a reason to lie awake tonight worrying. Sleep anxiety itself, the stress of worrying about whether you are sleeping well enough, is one of the most common causes of poor sleep, and it is a particularly cruel irony. The knowledge of what sleep deprivation costs is not a weapon to turn against yourself. It is context for understanding what sleep provides. And you are providing it right now. The simple fact of being here in the quiet, in the dark, allowing rest to come, is already performing the function. Every night that you sleep well is a night that the river flows, the proteins are cleared, the molecular balance is restored. Every night of good sleep is, in a very specific and biological sense, an act of care for the long-term health of your mind. You are, right now, taking care of your brain in the most fundamental way possible. There is one more dimension of the sleep deprivation story worth considering, because it connects the glymphatic science to something that most people experience very personally: the feeling of emotional heaviness after a poor night's sleep. Most of us have had the experience of waking up after too little rest and finding that the world feels slightly harder, the day slightly more burdensome, the challenges slightly more overwhelming than they would otherwise be. We tend to attribute this to tiredness, and we are right, but the specific neurobiology behind it is more precise and interesting than just fatigue. When the glymphatic system does not run fully, when the molecular environment of the brain's interstitial fluid is not adequately maintained, the impact is not limited to the accumulation of large proteins like amyloid beta and tau. The interstitial fluid also contains smaller, rapidly acting molecules, neurotransmitter precursors, metabolic byproducts of synaptic activity, inflammatory signaling molecules. The precise balance of these molecules affects the tone and responsiveness of neural circuits in ways that are subtle but real. The prefrontal cortex, the region most associated with higher-order thinking, with the ability to hold multiple considerations in mind simultaneously, with the regulation of emotional responses, with the capacity for long-term thinking rather than short-term impulse, is one of the regions most sensitive to sleep loss. When the molecular environment of the prefrontal cortex is disrupted by inadequate clearance, when the arousal systems that support its normal functioning are running at reduced capacity because the chemical housekeeping has been incomplete, this region operates less efficiently. The inhibitory control it normally exerts over more reactive, emotionally driven regions of the brain is diminished. The amygdala, freed from some of the prefrontal cortex's normal oversight, responds more intensely to threatening or negative stimuli. Researchers at the University of California, Berkeley, who have studied the effects of sleep on emotional brain function extensively, have described this as a kind of overnight therapy that sleep provides. A process by which the emotional brain is regulated, the reactions of the day are processed and proportioned correctly, and the neural circuits are reset for the balanced emotional functioning of the next day. When sleep is disrupted, this overnight therapy is incomplete, and the emotional brain, left without the molecular restoration and the sleep-dependent circuit recalibration, enters the new day in a slightly heightened state of reactivity. This is why the same traffic jam, the same difficult email, the same minor conflict with a colleague that you would handle with ease on a well-rested day can feel genuinely overwhelming after a poor night's sleep. It is not that you have become weaker or less capable. It is that the specific biological processes that maintain your emotional resilience, the molecular clearance, the circuit recalibration, the neurochemical restoration, have been shortchanged. Your brain's emotional regulation is a function of its maintenance, and maintenance requires sleep. There is also something important to say about the immune system, because the glymphatic system does not operate in isolation from the brain's immune environment. The brain has its own specialized immune cells called microglia, and there is growing evidence that glymphatic clearance and microglial function are intertwined. The clearance of inflammatory molecules and protein aggregates from the interstitial fluid depends partly on glymphatic flow, and when that flow is impaired, inflammatory molecules accumulate at higher concentrations. Elevated neuroinflammation, in turn, can impair the function of astrocytes, the cells that form the channel walls of the glymphatic system and maintain the aquaporin 4 channels in their correct polarized configuration. Impaired glymphatic function leads to neuroinflammation, and neuroinflammation further impairs glymphatic function. Another circular relationship that illustrates how deeply the health of the sleeping brain is interconnected. Sleep, then, is not only a clearance system but a maintenance system for the brain's immune environment. Each night of good sleep reduces the inflammatory burden in brain tissue, allows microglial cells to reset to their normal surveillance mode function, and maintains the conditions that support optimal glymphatic efficiency the following night. The system is self-reinforcing in both directions. Good sleep begets more effective sleep, and poor sleep makes the next sleep less restorative. Understanding all of this, the molecular, immune, and circuit-level consequences of inadequate sleep, is not meant to generate anxiety. It is meant to illuminate the deep biological logic behind something that most human cultures have always known intuitively: that sleep is sacred time, that it should be protected, that giving the body and mind the rest they require is not laziness or luxury but maintenance of the most essential and irreplaceable equipment you will ever possess. You are providing that maintenance tonight. Let us come back to you, to this specific moment, to the room you are in, the weight of the pillow, the temperature of the air around your face, the sounds of the night outside, traffic or wind or rain or silence. You are here, in this moment, at rest, and inside your skull, something quietly extraordinary is taking place. Your locus coeruleus, that small, ancient cluster of neurons deep in your brainstem, is slowing down. The norepinephrine is withdrawing. Like the lights of a city dimming late at night, one by one, the alert chemicals of wakefulness are fading. And as they fade, a kind of opening is occurring in the landscape of your neural tissue. The spaces between your neurons, those microscopic channels of extracellular fluid, are gently widening, the cells pulling back slightly, the tissue becoming more permeable, more accessible, more ready. And somewhere at the base of your brain, in those intricate folds of the choroid plexus, fresh cerebrospinal fluid is being produced at this very moment. Silently, continuously, like a spring that never stops, clear and clean and carefully balanced. It forms in the ventricles and flows outward through the chambers, emerges into the subarachnoid space, and begins its inward journey along the periarterial channels. The arteries of your brain, those vital, flexible vessels, are pulsing slowly, not with the rapid, urgent beat of waking circulation, but with a slow, rolling vasomotion driven by the oscillating norepinephrine from your brainstem. Roughly once every 50 seconds, a gentle constriction moves down the walls of the periarterial channels, and with each constriction, a pulse of cerebrospinal fluid is nudged deeper into your brain, like the slow squeeze of a soft pump, like the patient, persistent action of a river working through stone. The fluid is moving now through your frontal lobes, through the tissue where the concerns and plans and ambitions of your day had their home. It is moving through the circuits where you weighed your decisions today, where you worried, where you imagined, where you hoped. It is moving through the hippocampus, that curved structure deep in the temporal lobe where new memories were laid down today, where the events of this specific day are being sorted and held. It is moving through the structures that process emotion, the amygdala, the anterior cingulate cortex, the insula, through all the places where feeling lives. And it is finding the proteins, the amyloid beta that was produced today by your neurons in the course of their normal energetic activity, the tau that floats in the interstitial currents, the smaller metabolic byproducts of a day's worth of firing and signaling and computing. The fluid is collecting them the way a slow, careful stream collects sediment. Not dramatically, not all at once, but continuously, patiently, until it is laden with what needs to be removed. And then it exits through the paravenous channels, through the meningeal lymphatics, toward the cervical lymph nodes in your neck. The waste your brain generated today is leaving your skull in that fluid right now, beginning its journey toward the lymphatic system and ultimately toward the organs that will process and eliminate it. By the time morning comes, and you begin to rise back through the stages of sleep toward waking consciousness, a substantial portion of what needed to be cleared will have been cleared. The molecular balance of your interstitial fluid will be better maintained than it was when you went to bed. The specific concentrations of amyloid beta and tau that accumulated during your waking day will be reduced. The chemical environment in which your neurons will fire tomorrow morning will be, in a very literal sense, cleaner than the chemical environment they fired in tonight. This is not a poetic metaphor. This is molecular biology. This is measurement confirmed, fluorescent tracer imaged, PET scan validated, peer-reviewed biological reality. Your brain is cleaning itself as you rest. There is something that moves me about this, something I want to spend a moment with, and that is the relationship between consciousness and this cleaning process. Every thought you have leaves a molecular trace. Every time a neuron fires to encode a memory, to process an emotion, to generate a plan, to recognize a face or a word or a feeling, that firing costs something. It consumes energy, and it produces metabolic waste. The act of being aware, of experiencing anything at all, has a molecular cost that must be accounted for. Sleep, then, is not merely the absence of consciousness. It is the accounting that follows consciousness. It is the settling of the molecular tab that waking life runs up. And there is something quietly beautiful about that reciprocity, the idea that the very activity of being alive and aware and present in the world generates the need for this nightly ritual of cleaning. That experience and rest are not opposites, but complements. That one requires the other. You could not have the day without the night. Not just because rest requires sleep, not just because memory consolidation requires sleep, but because the molecular residue of experience requires nightly clearance or it becomes a burden. Consciousness, at its most fundamental biological level, is a process that must be cleaned up after regularly. And sleep is the nightly act of that cleaning. Think, if you can hold a thought gently without being drawn too far back toward wakefulness, about all the nights that your glymphatic system has run for you. Since the day you were born, across every night of your life, in childhood, in adolescence, through all the seasons of your adult years, every night that you slept, the river has flowed. The interstitial space has expanded. The norepinephrine has oscillated. The CSF has moved through the channels, collecting what needed to be cleared, and carried it away. You did not know this was happening. No one knew this was happening until quite recently. But it has been happening for every night of your entire life, with the same quiet reliability as your heartbeat, as your breathing, as every other automated maintenance function your body performs without asking for your attention. Now you know, and knowing, I hope, creates not anxiety but wonder. Not worry about whether you are sleeping correctly, but appreciation for the extraordinary sophistication of the system that is running inside you right now. You are carrying a cleaning system that cost evolution hundreds of millions of years to develop. A system that uses the physics of fluid dynamics and the chemistry of water channels and the neuroscience of slow oscillations to maintain the molecular cleanliness of the most complex object in the known universe. And it requires nothing of you except that you rest. Tonight, that is exactly what you are doing. The glymphatic system does not operate in isolation from the rest of your life. It is embedded in the biology of aging, shaped by your lifestyle, influenced by your habits in ways that researchers are only beginning to fully map. Understanding these connections adds texture to the picture, and, importantly, suggests that the decisions we make during our waking hours have a real and measurable impact on the quality of what happens while we sleep. One of the most important things to know about the glymphatic system is that it declines with age. This is not inevitable in the sense of being immune to intervention, but it is a well-documented biological reality. In aging animals, and in human studies where indirect measures of glymphatic function have been assessed, the efficiency of glymphatic clearance decreases progressively from early adulthood onward. Several mechanisms contribute to this age-related decline. The aquaporin for water channels in astrocyte end feet become less precisely organized with age. The tight clustering of AQP4 channels at the perivascular surfaces, a configuration called polarization, is gradually lost. Without proper polarization, the channels are still present but distributed less specifically, and the efficiency of fluid exchange across the astrocyte end feet membrane is reduced. Studies in older mice have documented a significant loss of AQP4 polarization compared to young animals, along with corresponding reductions in glymphatic clearance. The blood vessels themselves change with age. Arteries become stiffer, less elastic, less capable of the slow rhythmic vasomotion that acts as the glymphatic pump. When arterial stiffness increases, the amplitude of the vasomotion decreases, and the driving force for CSF flow through the periarterial channels is reduced. Conditions associated with accelerated vascular aging, hypertension, diabetes, atherosclerosis are therefore also associated with impaired glymphatic function. This may be one of the mechanisms through which cardiovascular risk factors increase the risk of dementia, not only through their direct effects on blood flow, but through their indirect effects on glymphatic clearance. Age-related changes in sleep architecture also contribute to the decline. As people age, they tend to spend less time in deep stage 3 sleep, the slow-wave sleep that is most critical for driving glymphatic clearance. The amplitude of the slow waves decreases. The total amount of N3 sleep per night diminishes. And since the glymphatic system's optimal function depends on the specific oscillatory dynamics of N3 sleep, a reduction in the depth and duration of slow-wave sleep directly translates to reduced glymphatic clearance. The convergence of vascular stiffening, AQP4 depolarization, and sleep architecture changes with age may explain why cognitive decline and dementia risk accelerate precisely in the age ranges where these biological changes become most pronounced. But the news is not entirely pessimistic, because researchers have identified several modifiable factors that appear to support glymphatic function. And most of them are familiar, accessible, and requiring nothing more exotic than thoughtful attention to lifestyle. Physical exercise, for instance, appears to improve glymphatic function, not directly during exercise, when elevated norepinephrine actually suppresses glymphatic flow, but through its effects on the subsequent sleep, which tends to be deeper and more restorative in people who exercise regularly. Regular exercise also improves cardiovascular health. Arterial elasticity, blood pressure regulation, metabolic efficiency, and through these effects, it may help preserve the arterial vasomotion that drives the glymphatic pump. Animal studies have directly observed improved glymphatic function in exercising animals compared to sedentary controls. Diet is another variable. Research has suggested that omega-3 fatty acids, found in fatty fish like salmon and sardines, and in plant sources like flax seeds and walnuts, may support glymphatic function through effects on the lipid composition of cell membranes and on the regulation of the aquaporin water channels. Whether dietary omega-3 intake meaningfully improves glymphatic clearance in humans remains an area of active investigation, but the mechanistic plausibility is real. Chronic psychological stress is a factor that appears to work in the opposite direction. Stress activates the norepinephrine system. The locus coeruleus fires more actively under stress, releasing more norepinephrine throughout the brain, and elevated tonic norepinephrine compresses the extracellular space and suppresses glymphatic flow. Chronic stress also disrupts sleep quality, further impairing the conditions necessary for glymphatic function. The effects accumulate. A stressed brain sleeps poorly, and a poorly sleeping brain clears less waste, and a brain accumulating more waste may find it harder to manage stress. This suggests that stress management, whatever form it takes for a given person, whether that is exercise, meditation, time in nature, connection with others, creative work, or simply adequate rest, may support brain health not only through psychological pathways but through the specific biological mechanism of preserving and supporting the glymphatic system. None of these lifestyle factors represent dramatic interventions. They are the familiar recommendations that appear in every guide to healthy living. What the glymphatic system adds to this picture is a specific mechanistic rationale for why they matter for the brain. Sleep well because the glymphatic pump needs slow-wave sleep. Exercise because it deepens that sleep and keeps your arteries elastic. Manage stress because chronic stress elevates the norepinephrine that suppresses glymphatic flow. These are not vague correlations in population data. They are specific biological processes with understood mechanisms. The glymphatic system is also a target of current therapeutic investigation. Researchers are exploring whether specific drugs or interventions might enhance glymphatic clearance by improving AQP4 function, by enhancing arterial vasomotion, by optimizing sleep architecture. Some early animal studies have shown that certain compounds can increase glymphatic flow, and there is cautious hope that such interventions might eventually be useful in preventing or slowing neurodegenerative disease. The path from animal studies to validated human therapies is long and uncertain, but the mechanism is identified, the rationale is sound, and the investigation is underway. You live in an era when the brain's cleaning system has been discovered, characterized, and is actively being studied by some of the most talented researchers in neuroscience. The understanding of what sleep does, specifically and mechanistically, is deeper now than it has ever been in the history of science. And that understanding, I hope, adds weight and meaning to the simple act of choosing to rest. We have traveled far tonight in the quiet. From the laboratories of Maiken Nedergaard at the University of Rochester, where fluorescent tracers first lit up the hidden rivers of the sleeping brain, through the choroid plexus and the ventricles where the CSF is born, through the periarterial channels and the aquaporin four gates and the wide, expanded interstitial space of the deeply sleeping brain, through the slow oscillations of norepinephrine and the gentle vasomotion of the arteries and the patient, rhythmic pump that drives the cleaning tide through your neural tissue, through amyloid beta and tau and the story of what happens when those proteins are not cleared, through the science of sleep position and the consequences of deprivation and the broader landscape of how a lifetime of choices shapes the glymphatic river's capacity to flow. And now we are here again at the quiet center of all of it. At this moment, at you in the dark at rest, something specific and beautiful is happening inside you right now, in the sleeping architecture of your brain. The norepinephrine oscillations are running their slow infraslow rhythm. The arteries are pulsing in their gentle vasomotion. The cerebrospinal fluid is moving through the channels it has traveled every night of your life since you were born. Inward along the arteries, through the tissue, outward along the veins, carrying the molecular residue of today toward the lymph nodes and the body's disposal system. The river is flowing. Right now, while you listen to this, or while you have drifted past listening into the soft interior of sleep. The river does not need your consciousness to run. It does not need your attention or your permission or your understanding. It only needs your rest. And rest is what you are giving it. There is something I want to offer you as a final thought before the words fade and the quiet takes over entirely. And that is this: the discovery of the glymphatic system changed the way we understand sleep. Not by making it more complicated or more demanding or more anxiety-producing, but by revealing that sleep is not passive. Sleep is not the dark, empty gap between the meaningful parts of your day. Sleep is where maintenance happens, where the molecular debt of consciousness is paid, where the most essential housekeeping of the self takes place. You are, in sleep, being cared for by your own biology, a system that evolution spent hundreds of millions of years perfecting is running inside you tonight. It asks nothing of you. It requires no effort, no discipline, no special knowledge. It only asks that you create the conditions in which it can do its work. That you find the dark, find the quiet, find the warmth and the stillness. That you allow yourself to let go. That is what you are doing right now. And it is enough. Tomorrow morning, when you open your eyes and take in the first sensations of a new day, the brain that greets the morning will be, in a very specific and biological sense, cleaner than the brain that lay down last night. The amyloid beta will be reduced. The tau will be reduced. The molecular environment around your synapses will be more precisely balanced. The extracellular fluid that bathes your neurons will be more carefully maintained. You will feel it as clarity, as readiness, as the particular lightness of a mind that has been given what it needs. Every night of good sleep is a gift you give to the version of yourself who will wake up tomorrow. And every night of good sleep is a contribution to the version of yourself who will be thinking and remembering and feeling and experiencing 10 years from now, and 20 years from now and beyond. The glymphatic river runs tonight, and it runs for the long life of your mind. Sleep well. The river is flowing. If you found tonight's journey through the sleeping brain worth taking, if the science made you feel something, or helped you let go a little more easily, I would be so grateful if you would subscribe to Deep Science Sleep. This channel exists for exactly this. For the people who find that learning quietly at the edge of sleep makes the night feel a little less ordinary and a little more like the extraordinary biological event that it always was. A like on tonight's video, or a comment telling me what you found most interesting or surprising, makes a genuine difference in helping others find their way here. And if you know someone who might appreciate this kind of slow, science-forward company in the dark hours, please share it with them. Every week we take another slow, gentle journey into the science of what happens when we sleep, when we dream, when we rest, the biology of the quiet hours. Next week, we will explore something equally strange and beautiful. I hope to see you there. Until then, wherever you are, whatever this night holds, sleep well. The river is flowing.